So we are now ready to move on to the fourth module, to the fourth inquiry question, which is genetic variation. How can genetic similarities and differences within and between species be compared? So we just looked at how essentially all of these processes are working at a molecular level, what is happening, so to produce these molecules and so on. So now we are going to look at essentially the genetic similarities and differences, and yeah, now we're looking at a more macro picture. So genetic variation essentially, now when it comes to traits, what happens is that a trait can be recessive or it can be dominant. A dominant trait will always be expressed over the other traits, and a recessive trait will only be expressed if there are two. It requires two of the same allele to be represented. So we denote a recessive trait with like small letters. So for example, A, that's a recessive trait, and a dominant trait is denoted with capital letters. So A. So the terminologies I'll have you remember here is that this is called homozygous dominant. So it's homozygous because we've got two of the same alleles, both are capital A's, and it's dominant because they're both dominant. They're both expressed. They'll be expressed. So anything that's capital when it comes to like denoting genotypes, it's going to be dominant if it's written in capital letters. So this is homozygous dominant. Homozygous dominant. Definitely jot that down. I cannot write it because there's not enough space and it's a long word. So homozygous dominant. Alright, ZYGOUS. So that's homozygous dominant. Now, if you've got two small ones, it's still going to be homozygous because they're the same, but they're going to be homozygous recessive. Now, you also see this, which is a. Now, if this condition is dominant, which it is in this case, it's going to be expressed over the smaller condition. Alright? So here, I want you to think of the fact that these A's, they represent a trait. Let's say they represent black hair color. So so the big A represents black hair color, and the small a represents red hair. Okay? So that means that red hair is a recessive trait. Black hair is a dominant trait. So over here, it's AA. So if someone has the genotype AA, they're homozygous dominant, they're gonna have black hair. Like that's their phenotype. The black hair will be the phenotype. It's what we see. The genotype is what their genes are. If they have two small a, so they are AA, they're homozygous recessive, that would mean they have red hair. Now, if they have a big A and a small a, that's their allele, right? Which one would they be? So remember, because A is a dominant trait here, it's going to overpower the small a. So they're not going to have red hair, they're going to have black hair because this A is overpowering the small one. So this is called heterozygous. So it's called heterozygous. And it's heterozygous because you've got two different alleles. That's why. So homozygous, two of the same ones, but homozygous dominant is when they're dominant. Homozygous recessive is when they're both small here, and this is heterozygous. It's a mix. So, how are these expressed? This is how they're expressed. A gene is allele is codominant if both alleles in a gene pair are fully expressed. If they're both fully expressed, they're codominant. If they're both partially expressed, they are incomplete dominance. And there can be multiple alleles. So a trait can be coded for by multiple alleles, not just two, but multiple different alleles can code for a gene. And we'll talk about what that means in a second. So I'll draw your attention to this part over here now. This is what your chromosomes look like. Okay, so this is essentially, remember, we've got so this is where the genes are carried on these 22 bad boys here, and then here you've got these two sex-linked chromosomes, your X chromosome and your Y chromosome. So hopefully that makes sense. So let's have a look now at what incomplete dominance and codominance and all of that is. So if we breed a red flower with a white flower, essentially what will happen is that incomplete dominance would be if you get a pink flower, so you know, both of them like do not fully compete with each other. Codominance would be if we get a red and a white flower that they both actually got through. So that's that. Now here we've got multiple alleles. What do multiple alleles look like? So multiple alleles essentially, as you can see, let's say this we denote C dominant as, so this is the allele C dominant, C to the power of CH, CH and C, small c. So the genotype, the genes, remember, because it's like two parents have to mate, so here the genotype can be CC. That will result in a wild type brown fur rabbit. CCH and CCH will result in a chinchilla, which is a black-tipped white fur. Now CHCH will result in the Himalayan fur, which is white fur with black paws, nose, ears, and tails. And finally, this recessive allele of homozygous recessive, it will result in an albino fur, which is white fur. So hopefully that makes sense. Let's move on then. Now, if you have any questions, please ask. Alrighty. So now we know how these are expressed. Now we're going to look at pedigrees. What are pedigrees? Pedigrees essentially are these images, these not graphs as such, but these these pedigrees help us basically trace the way in which the way in which phenotype has sort of passed through generation. Remember, we can't trace the genotype without like actually doing like gene sequencing and stuff. We can't determine what the genes are, but we can see the phenotype. Phenotype, remember, are the things we observe, like hair color, eye color. It's the phenotype. It's things we can see. All right. I might think of pheno as like face. You can see it. All right, a very very basic example, but maybe one way to think about it. But remember, it does not only have to do with face, it can also have to do with like different conditions that are passed in family, like different diseases and stuff as well. That was, like I said, very basic. But yeah, look, it helps us see how phenotypes are being traced through generations. We always want to include a key, and you want to label generations for ease of reference. Now, remember we talked about recessive versus dominant traits. Now, if a trait is recessive, it will skip generations. It's going to skip generations if it's recessive. So this is, the way you read it is that this is the first generation. Alright, so the squares are males, the circles are females, and any circle that's colored means that the person is affected, like they are affected with whatever the trait is. These are the people. Just because they're not circled doesn't mean that they don't have it. It could mean that so if they're not circled, it means two things. First things means that they obviously don't have the trait. Second is that they could be a carrier, so they can carry the trait on one of their genes, but they may not have it. So that's that. So this is the first generation, second generation, third generation. Now when you see the two lines connecting to, like, for example, these three here, so that means this couple has two daughters and one son, and so then the lines outside are like the partners of the family. Alrighty, so a recessive trait will skip generations. In this image, we can see that the recessive trait, we can see it's not a recessive trait, that everyone in the three generations has it. So it's not everyone, sorry, but someone, at least one person in each generation has it, so it's not recessive. That's how you tell recessive versus dominant. Dominant does not skip generations. It can't skip generation because it's dominant. All right, so that's why we can say that safely that this is a dominant trait. Now, how do we tell if it is sex linked versus if it is autosomal? So, autosomal traits are carried on these 22 genes here. That's autosomal. They're carried on the body cells. Sex linked are carried on the sex cells right there. So essentially over here, what we can see is that sex-linked traits will affect one sex more. All right, so that's important. So with autosomal traits, so that's the key here. So sex-linked traits affect one sex more than the other sex. So if we see here, we can see that it is affecting the women in the family and the men in the family pretty much the same. So that's one key to say that it's actually going to be an autosomal trait, not a sex-linked trait. All right, that's one way to tell it. Another way to tell if a trait is sex-linked is if it goes from the mother to the son. And that's because the son, so males have XY chromosomes, females have XX chromosome. So because males have XY chromosome, they get their X chromosome from the mother, right? Because they get their X chromosome from the mother, it means that basically if the mother is a carrier and the trait is sex linked, it's on the X chromosome, the child is going to get it. The son is going to get it. The daughter may not get it because the daughter gets two X chromosomes, one from the father, one from the mother. So she could be a carrier, but she may not get it. But if the mother is in fact is a carrier of the condition, the son more than likely, if it's sex linked, the son's going to have it. That's another way to sort of say that it is sex linked. So with this, with determining pedigrees, practice is the best way to go. There's no sort of rule written in sort of in hardcore rules, but it's these little patterns that help you determine which one's which. So recessive dominant, pretty easy to tell apart, because if it's recessive, it's not gonna it's gonna skip a generation. If it's dominant, it will not skip a generation. Can't skip a generation. Probably a bit harder to tell between sex linked and autosomal, but use these indicators. Like if it's sex linked, it generally affects one sex within the pedigree more than the other, and also you'll see that it generally, if it's sex linked, it generally there's a pattern of it going from the mother to the son. Like that's very obvious. So let's move on now and let's have a look now actually at Punnett squares. This is how we now theorize how genes pass. This is how we actually theorize how genes pass from the parent to the offspring. Here we go. So this is how we generate potential genotypes and phenotypes. So let's have a look at this. Let's say we are going to determine the petal of a flower here, like what color the flower is going to be, and we know that this is the genotype of the parents. All right, the pollen is, what is it going to be? What word did we use for it? And I'll give you three options. Is it going to be homozygous recessive? Is it going to be homozygous dominant? Or is it heterozygous? What is it? What is capital B and small b? Give you 2 seconds. Oh no, not 2 seconds. Let's give you 10 seconds. You ready? 5, 4, 3, 2, 1. Alrighty. So if you said heterozygous, you are correct. This is going to be heterozygous because you've got a capital B and a small b. So they're not same. So the alleles are not the same because they're different, because you've got a dominant and a recessive allele. So the pollen is BB, and so is the pistil. All right, those are the sexual organs of a flower. So now, essentially, not sexual organs. Sorry, those are the sexual elements of the flower. That's right. So that's where that they're gonna play, like their specific genotypes are going to play a huge role in the phenotype of the flower. So now we just cross it over basically. So we go BB is going to give us this. BB, now this is homozygous dominant, remember? that this trait is going to be heterozygous. It's still going to give us the color purple. Why? Why will it give us the color purple? Because of the fact that it's a dominant trait. Because purple flowers is a dominant trait. So purple's going to win over the white here. Over here, it's big B and small b. Again, same thing. Because the purple flower is the dominant trait and you've got an allele for the dominant trait and the recessive trait, the dominant trait's going to win. But you've got the small b and the small b here. So potentially you could get a white flower because it's both recessive alleles. So that can happen. This is how we determine the phenotype, the genotype, potential genotypes. So this is now, we can say the phenotype, there's a 3-to-1 ratio that it's going to be purple flower to white. So that's the ratio of purple to white is 3-to-1. And so the genotype ratio is 1-to-1. So there's 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So whenever you make these Punnett squares, you want to give a key, you want to talk about parental phenotypes and genotypes, then fill in the Punnett square, and then give the percentages of the ratio. So here, the percentage would be that it's 75% purple versus 25% ratio of white flowers, if that makes sense. So if you have any questions, please ask. Remember, no question's a silly question. Hopefully we're now feeling a lot more confident with this, and if you, like I said, have any questions, just let me know. Let's move on then.